Swayam Prabha. Digital India. Educated India.
Welcome to lecture 20. This is the NPTEL online certification course on bioreactors. In the previous two lectures, we had looked at bioreaction stoichiometry as one of the means of getting some insights into what is happening at the cell level in the bioreactor. The cells in the bioreactor are the actual factories. We worked out a problem in the previous lecture. Now let us go forward with the module itself, the module 5. We are going to begin talking about windows to the cell. Now, we said we will first consider it as a black box, the cell, and then we'll open up windows and look through the windows to see what is happening inside the cell. So, the windows to the cell are the indicators of cellular status of the cell or indicators of metabolic status of the cell. These terms, cellular status, metabolic status, energy status, and so on and so forth, redox status, and so on, these are kind of soft terms. You should not take them too strictly. These are kind of indications of what could be happening, and these are kind of common terms that are used. They are not, of course, fully understood, but they are useful. The cellular redox status is a well-known indicator, and cellular energy status is another well-known accepted indicator of the cellular or the metabolic status of the cells. The cellular redox status is typically derived from NADH. NADH, nicotinamine adenine dinucleotide, and the hydrogenated form of that. This conversion takes place in various metabolic reactions or accompanied with various metabolic reactions in the cell. NAD plus H plus giving you NADH. So, this has the reducing power which is taken forward for so many different important functions in the cell. So, the redox ratio, one of the ways of looking at it at least, is the ratio of NADH concentration to the total concentration of NAD plus and NADH. We have already seen that NADH fluorescence can be used to monitor the cell concentration, because the intensity of NADH fluorescence can be directly linked to cell concentration. Here it is redox ratio, the NADH divided by the total NADH pool, which consists of NAD plus and NADH. Okay, this is the redox ratio. Similarly, or in some situations, the ratio of NADH to flavin adenine dinucleotide, another important such molecule, is also taken to be the redox ratio. For energy status, it's somewhat a little more straightforward. We all know the energy is made or energy currency of the cell, ATP, adenosine triphosphate, is made by the phosphorylation of adenosine diphosphate. So, the ratio of ATP to the pool, ATP plus ADP, sometimes AMP is also added here, but let us just look at these two. ATP divided by ATP plus ADP is taken to indicate the cellular energy status. So, a lot of measurements are made on these, correlated to the happenings inside the cells, correlations with respect to what is important from a bioreactor context, and efforts are made so that these indicators are varied in a particular fashion or kept constant at a particular known value to achieve desired performances by the cells in the bioreactor. So, that is one more step towards understanding what is happening at a cellular level. Sometimes even ATP by ADP is considered the cellular energy status. There are various ways of defining this. I've just given you a couple of them. What one needs to understand is that very many metabolic processes contribute to the above. Okay, so it is not a one-to-one kind of a relationship. So many things contribute to redox status, so many things contribute to energy status. So one needs to have some level of interpretation, or one needs to interpret this in that kind of a fuzzy fashion. It's more of an indicator. You may want to look at this paper, which has also been given in your notes as one of the papers that you would want to look at. Let me direct that to you. This is number 30, NADH culture fluorescence. The authors are Shepper, Gebar, and Shugal. This is the paper for NADH culture fluorescence. The title of the paper itself is Monitoring NADH Dependent Culture Fluorescence During Cultivation on E. coli. It was published in Chemical Engineering Journal in 1987. This is something that gives you some relationship to the culture energy status. Let me, I do have a paper here. Yes. Let me briefly tell you how this is done with energy, with NADH status. To very briefly read the abstract, NADPH dependent culture fluorescence was monitored during various E. coli cultivation processes, batch and continuous cultivation, in stirred tank reactors or bubble column reactors. Various strains of E. coli, including genetically modified E. coli, were used. Measurement of fluorescence signal allows biomass estimation under certain conditions, this we have already seen. The monitoring of culture fluorescence is useful for obtaining information on the metabolic status of cells. The effect of cells of different substrates, glucose and oxygen, was studied. The measurements were performed in synthetic and complex media, and the influence of the composition on of each medium was investigated. A microflurrimeter was used as a routine sensor for cultivation control and provided insight into the status of cells. Okay. The what I'd like to point out is this. You see, this is biomass versus time. The points here are the individual points. Let's increase the size a little bit so that you can see it a little better. Right? Yeah. The points are experimental points, and the line is given by culture fluorescence. You see a very nice relationship. This is what we talked about as a measure of the cell concentration itself. This is biomass or cell concentration in grams per liter versus time by two methods. One is by the traditional the OD method, and the other one is by the culture fluorescence method. Okay. It agrees in many situations. It doesn't agree in one such situation. This is bubble column conditions. If it is a stirred reactor, there is absolutely no problem, whereas in a bubble column there seems to be interferences which lead to a difference between these two. So one should be careful while using culture fluorescence in bubble columns. It makes sense because bubbles will interfere. And then one more thing I'll point out from this paper. Here. You see this figure? Let me blow it up a little bit if you want. Yes. This is culture fluorescence versus time. The fluorescence was at a certain level when oxygen was used, when oxygen in air was employed. Okay, let us read the figure 7 is what it just says. Okay, that doesn't matter. You have culture fluorescence at a certain level. When nitrogen is introduced, within a few seconds, the culture fluorescence goes up. The anaerobic status goes up. When nitrogen is employed, the cells become anaerobic because oxygen is no longer available as final electron acceptors, right? So the culture fluorescence at an anaerobic state is much higher than the culture fluorescence as an aerobic state, because you'll find NADH forming more. So this very clearly indicates the aerobic state of cells. When oxygen is turned back on or air is turned back on, it comes back to its original level. So this is a nice indication of what is happening inside the cell just by measuring the culture fluorescence. That's what I had talked about earlier. You can read this paper if you are interested. For the purposes of this lecture, I think this is good enough. Okay. In the case of okay, so that is using culture fluorescence. Now let me talk about some of our work which was done quite a while ago, but it also is on the same lines as this. This is measurement of intracellular pH. This is pH inside the cells. This is distinct from the pH of the medium. pH of the medium we know goes up and down because of acid-base additions, and we said that the pH needs to be controlled at an optimum value for best bioreactor performance and so on and so forth. We are talking of intracellular pH inside the cells. Now the pH inside the cells can somewhat be related to the energy status of the cells, because there's something called a Mitchell's hypothesis. In fact, Peter Mitchell won the Nobel Prize for this hypothesis. This essentially says that a hydrogen ion gradient across a membrane, intact membrane, is what provides the energy for ATP formation from ADP. So in some sense you can link the energy status of the cell to the intracellular pH level. Okay. There are some fluorescent dyes that indicate the pH through their fluorescence by getting inside the cell and so on, but we couldn't really use them because those dyes leaked out. BCECF-AM is one such dye. There's of course some detail, it might be of interest to some, so that's why I'm mentioning it briefly. These dyes could leak out, so we used a dye called 9-aminoacridine, which is a weak base whose distribution depends on the difference between the extracellular and the intracellular pH. There are also these processes take place across the plasma membrane in lower organisms such as bacteria and so on. So that is what we were using. So what we did was we developed this method of intracellular pH measurement online continuously in a bioreactor, and then we used intracellular pH to grow cells at a particular intracellular pH, which is indicative of energy status, after studying that different intracellular pH levels indicate different metabolic conditions in the cell. So what we did was we measured intracellular pH through fluorescence, and we found that glucose addition, the addition of the substrate, can modify intracellular pH. So the glucose additions were done so that the intracellular pH was maintained at a certain higher level compared to the base level, so a higher energy level. And when we did that, we could reduce the anaerobic product formation by about 4-fold. So if it is anaerobic growth, then the outcomes are much less than the aerobic growth in terms of cell yield and so on. So we were able to do that much better, and we could reduce the anaerobic product yield. So these things could also be done. There are a couple of papers on intracellular pH which are given here. I would not discuss them further because of the introductory nature of this course, but if you are interested, you can look at these two papers given under 31 and know more about the intracellular pH itself. Okay, let us move forward from here. So the control, this is what I mentioned earlier. I've given this as a diagram here. Intracellular pH was controlled using fluorescence. This is not a pH probe. pH probe will measure only the pH of the medium. then transmitted, and the controller was the one that determined the glucose feed, whether it needs to be fed, or the feed rate of glucose. So that became the manipulated variable to maintain the intracellular pH at a certain set point to reduce the anaerobic product formation. So this is the control schematic of the whole thing. Okay. So now let's start looking inside the cell through various windows, doors, and so on and so forth. I'll tell you one example of such things, of looking inside the cell, and that is called metabolic flux analysis. Okay, we'll spend some time on this, the metabolic flux analysis. It's interesting. It became popular in the early 1990s. The method was known from the 70s onwards, 1970s onwards. It became popular when Valeno and Stephanopoulos published their paper on using metabolic flux analysis to provide them with directions to improve lysine production. Lysine is an amino acid that animal cells cannot make, and therefore, but that amino acid is required for a protein, so we need to supplement it through diet or other means, and therefore lysine is a huge product. Large amounts of lysine are produced annually, and therefore it's commercially a very important product. So if you improve the lysine production a little bit, then the profits could be high, or the price could further be brought down, and all these made the use of metabolic flux analysis a popular one in the early 90s, and a lot of work went on for the next 20 years or about 15 years on metabolic flux analysis. We will look at the principles of metabolic flux analysis next. So as an introduction, cells make products as a result of hundreds of reactions that take place inside them. You already know this. To improve product yields, the chemical reactions of the metabolic pathways can be targeted. And let us look at a method of analysis of those pathways toward improving product yields. Okay, this is our main aim. We are going to look at a method of analysis toward improving product yields, and that method is called metabolic flux analysis, or MFA for short. To know what metabolic flux analysis is all about, let us start with this. Let us represent all the cells in culture by a single large cell to begin with. Okay, so the volume of the cell equals the volume of all the cells in the bioreactor. The remaining volume is the broth volume. This could probably be less than 10% of the, even in concentrated situations, it would be about 10 to 20%, right, of the total volume. And let us consider these set of reactions that are occurring in the cell, a hypothetical set of reactions, but these are good to understand the principles of metabolic flux analysis. SO is a substrate that's outside, that goes inside and gets converted to S through probably the reaction R0. Then S could get converted to A or B. It gets converted to A at a rate of R1. This R1 is moles per time, and it gets converted to B at the rate of R2 moles per time. B gets converted to D at the rate of 4, which comes outside. A gets converted to C at the rate of R3, which also comes outside. Okay. So this is the situation here. Let us say that this is some important metabolic pathway that is happening in the cell. This is the case. It's good to note that S, A, B are inside the cell and therefore are intracellular metabolites, whereas S, O, C and D are extracellular metabolites. And metabolite flux is a common term that is used. The term flux is very different from the engineering term flux. The engineering term flux is amount transferred per unit area perpendicular to the direction of transfer per unit time. That's a flux, right? In the case of metabolic flux, historically moles per time, or in other words a rate has been used, an amount rate has been used to indicate flux. We will continue using that because it's widely accepted in the literature. It is a rate and not flux as in engineering terms. This you need to keep in mind. So the typical units of R are 0, R1, R2, are millimole per gram cell per time. Note that it is normalized with respect to mass and not volume, right? Rather, it's expressed in terms of mass, and it has not been normalized with volume. Now let us consider this large cell as a system. Let us write a balance on intracellular metabolites first. I'd like you to note that if we are writing balances on intracellular metabolites, we consider the cell as our system. This is our system, and we write the balances on the metabolites inside the cell. Let's begin with this S here. So rate of input of S minus rate of output of S, this is a system, that's what we are talking about, plus the rate of generation of S minus the rate of consumption of S equals the rate of accumulation of S. Here you need to, if S0 has come in as S0, then we consider it as transport across the system boundary. These two terms, input and output, have relevance, as you know, only when there is transport across the boundary of the system. So if S0 has become S0 here, then we can consider it as the input term. Otherwise, we consider it as a reaction term, which could be accounted for in generation or consumption. But we need to make sure that we count it only once. We cannot count it as input as well as generation, even if you want to follow a different strategy for doing this. If you follow this strategy, that if S0 becomes S0, then we consider it as transport. If S0 becomes something else inside, then it becomes a reaction. Similarly, A going to C, we are going to consider it as a reaction because A has become C. A has not become A outside. Then it can be looked at as transport. The main thing is that there should not be any double counting. So there is, if you look at that, there is no input of S into the cell. There is no output of S into the cell. That's not going anywhere. Whereas there is definitely a rate of generation and a rate of consumption. The rate of generation is R0. The rate of consumption is through these two reactions, right? Therefore, minus of R1 plus R2, that becomes minus R1 minus R2, and that equals ds dt. Similarly, for A and B, A is formed at the rate of R1, it gets converted at the rate of R2. So generation is R1, consumption is R2, R1 minus R2 is dA dt. Similarly, R2 for B formed at R2 and getting consumed at R4. Therefore, R2 minus R4 is dB dt. Now, there is a trick here. Okay, I want you to pay some attention. We are going to consider the surroundings of the cell as a system. Okay. When we focus on extracellular metabolites, we will consider the surroundings of our cell as a system and write our balances. That's what turns out to be consistent with our approach here. So if we write a balance on the extracellular metabolites, first on S0, S0 is getting out of the system here. This is our system now. Okay, it's getting out of the system. Therefore, there is no input, but there is an output, and therefore minus R0, which comes from here. It is not being generated, it's not being consumed. So minus R0 is ds0 dt. Similarly, for D, it is being produced here. The generation term alone exists. There is no input, there is no output, there is no there is no, in the case of the earlier one, there is no input, there is no output, there is no generation of S0, there is consumption of S0 through the reaction R0, which takes it to the other side, and therefore R0 is the consumption term. Here I have just lined up R0, R1, R2, that's the reason why R0 is here, and it is actually the consumption term. Therefore minus R0 is ds0 dt. You need to be a little careful with these things. Then the consumption term for C is R3, therefore R3 which is the consumption term, the other terms are 0, is dc dt, and R4 again a consumption, or I'm sorry, I'm getting a little confused here. Okay, this is for R3, this is our system, right? Yeah, I got confused between the systems. That's the reason I made that error here. You need to be a little careful. This is our system here. C is the extracellular metabolite inside its system now. This is being generated at the rate of R3, and therefore this is the generation term. So that is what is R3 here. Ri, Ro and Rc are 0, and therefore it is plus Rg equals dc dt. Similarly, for D, this is here, it is being generated at the rate of R4 by this reaction. Input, output and consumption terms are 0, therefore plus R4 equals dd dt. Okay. So this is the way you go about it. Once you get used to it, it's simpler, but you need to be a little careful. You cannot make errors like I inadvertently made by switching the systems in my mind. Be a little careful. So now the reason for me writing it this way is that, can you see a matrix evolving here? Now I've just lined up R0s, R1s, R2s, R3s and so on. So the set of these equations can be written in a compact form as this. Our first equation, we'll write it in this order: S0, S, A, B, C, D. Our equation, the way we do matrix multiplication, right? This term into this term, plus 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 this term into this term equals this term. So minus R0 is, the other terms are all 0 for the first term. Minus R0 is d dt of S0, which is what this one says. Minus R0 is d dt of S0. I'll show you one more thing. R0 minus R1 minus R2, the rest of the terms are 0, equals d dt of S. If you remember, that was the first equation. R0 minus R1 minus R2 is ds dt. One more equation just for completeness, and then we'll go forward. This first term is 0, R1, R2 is 0, R1 minus R3 is d dt of A. R1 minus R3 is d dt of A, right? That's the way the representation happens in a matrix form. Therefore, this matrix represents the set of equations that we got by doing material balances on the various metabolites in the cell, extracellular and intracellular. The number of equations would equal the number of metabolites, as we have seen, and that would equal the number of rows that we have here, right? Here we have 1, 2, 3, 4, 5, 6, and we had 6 metabolites, S0, S, A, B, C, D, so 6, and therefore that equals the number of rows. And number of rates would equal the number of columns here. There are 5 rates and 5 columns, R0, R1, R2, R3, R4, and there are 5 columns here in this matrix. So this is what is called a stoichiometric matrix, S with a hat above, stoichiometric matrix, multiplied with the rate vector or the rate matrix. R is d dt of the state variable, as it is called, state variable matrix x hat. Okay. So this is a compact representation of the entire set of equations, and this kind of a representation becomes much easier even if you have hundreds or thousands of equations. It's of course cumbersome to convert them into a computer program, but once you have converted them into the computer program, you can do all sorts of modifications if you look at it in this form. So that's the reason why we got it into this form. Reaction rate vector, and x is the state vector, vector of state variables. In other words, the metabolites are state variables. Okay. This is an important concept now. We had looked at something called a pseudo steady state approximation earlier. Let us revisit that. We talked about the rate at which a bolt is made, a bolt made every 5 seconds, and an engine, a car engine, made 1 every hour. We said since this is made every 5 seconds and this is made every hour, even if it takes 7 seconds or 3 seconds or 4 seconds to make a bolt, that variation in time, the unsteady state, unsteady nature of this process will not affect the nature of engine making. Okay, because this process is very fast, this process is very slow. If we focus on the slower process, then the much faster process can be assumed to be at steady state, whether it is actually at steady state or not. That is the pseudo steady state approximation. That's what is given here. If our interest is in engine making, about an hour characteristic time, the unsteady aspects of bolt making, a few seconds, are irrelevant. Thus, bolt making can be considered to be at steady state compared to the time scale of our interest, irrespective of whether this has been at steady state or not. So you need comparison. So in comparison with the time scale of interest, much faster processes can be assumed to be at steady state, and this is called the pseudo steady state approximation. We are revisiting this. This is an important concept, a little difficult to appreciate in the beginning, but once you get a hang of it, it becomes very powerful. Similarly, if we look at the intracellular metabolite concentration change, it happens over seconds, whereas growth typically happens over, let's say over characteristic times of hours. So this is a slower process growth compared to the process of intracellular metabolite concentration changes. So in comparison to the growth processes, if our interest is in growth related processes, the intracellular metabolites can be assumed to be at steady state. Okay, so this is the pseudo steady state approximation, and now see how it simplifies our approach. All the rate derivatives, the time derivatives of intracellular metabolites can be set to 0 by the pseudo steady state approximation if we are interested in growth related processes, which we are, growth and product formation. In the case of a bioreactor, definitely we are interested only in such processes, and in such a case we can directly take the intracellular metabolite variations with time to be 0, or this process to be at steady state. If we do that, then the intracellular metabolites are ABC. They can be directly put to 0. Okay, the other rates are extracellular metabolites. Let them be there. If this is the case, then you could split up this matrix as related to extracellular and intracellular matrices, because the intracellular metabolite related matrices will vanish. Okay, I've just taken R0, R3 and R4 and written the stoichiometric matrix from this part, which is relevant to that, to give us d dt of S0 C and D, and this is for the extracellular part. The extracellular part is this -1 0 0 0 1 0 and 0 0 1, corresponding to R0, R3 and R4, and they correspond on this side to d dt of S0 C and D, whereas intracellular, which corresponds to A, B and C, which are actually 0 here, can be taken from this particular from the ABC part of the matrix here, this particular part of the stoichiometric matrix. So I've represented this as a matrix that represents extracellular metabolites and a matrix that represents intracellular metabolites. You can take a look at the details and convince yourself with time. Let us move forward now. So the metabolic flux analysis is something like this. What all can be done with metabolic flux analysis? Right? This stoichiometric matrix gives us the various rates. The stoichiometric this representation gives us stoichiometric matrix, the various rates, and the rates of accumulation. Okay. So let us see what all can be done, or what are the common things that are done with such a representation, which would be useful for us from a bioreactor point of view. So estimation of intracellular metabolite flux from extracellular metabolite rates. Extracellular metabolites can be measured by some of the methods that we talked about in an earlier module, in module 3. So by measuring those, we can get an estimate of the intracellular metabolite flux, right? The rates at which things are going on inside the cell. Identification of branch point control nodal rigidity in cell pathways, this can be done. Identification of alternate pathways can be done. Analysis of fluxome, metabolome, and many omes can be done. Ome is nothing but a complete collection of everything that happens. A genome, for example, is a complete representation, collection of all the genes in the cell. Fluxome is a complete representation of all the fluxes in the cell, and so on and so forth. Okay. You can analyze that using this approach. We are going to look at only the first two as examples of what can be done with metabolic flux analysis. So let us look at the first thing, estimation of intracellular metabolite flux from extracellular metabolite rates. This is the same figure that we saw earlier, S naught going to S, which further goes to A and B, A goes to C and B goes to D. S naught, C and D are extracellular metabolites, S, A and B are intracellular metabolites. So we are going to measure S naught, C and D and then get the rates that are involved here. How do we do that? We know that the extracellular, the formulation corresponding to extracellular metabolites is this. We had already seen that earlier. Minus 100, 010, 001, R0, R3, R4. These are the rates that correspond to extracellular metabolites. Is d dt of S0, C and D. The equations corresponding to extracellular metabolites have been written in this matrix form. And this is the intracellular one. If S0, C and D can be measured, you can calculate these, right? You take points at reasonably closely spaced time intervals, then you can actually get the rates of variation of S0, C and D in the extracellular space. Knowing this, knowing the rates of S0, C and D, let us say our interest is in estimating R1 and R2. That's our interest. That is what we said here, estimation of intracellular metabolite flux or rates from extracellular metabolite rates. R0, the substrate uptake rate, can be estimated from S0 measurements. That's okay. Directly gives you R0. R3, R4, the metabolite or the product formation rates can be estimated from C and D measurements respectively. The intracellular matrix equation has 3 equations and 5 unknowns, right? Number of equations, number of unknowns. You know the degree of freedom concept. The degrees of freedom is the number of independent variables that are needed to be known to solve this set of equations completely. We have 3 equations, 5 unknowns. So 5 minus 3 is 2. So we need 2 of the 3 extracellular fluxes to determine R1 and R2. Let us say that those 2 are R3 and R4, which are known, which can be known from C and D measurements. We take measurements of C and D concentrations at reasonably closely spaced times, and we have these concentrations, we have these rates as derivatives, C2 minus C1 divided by T2 minus T1, and so on and so forth. So this is the matrix here, the intracellular matrix, which we have taken to be 0, which we had represented as, which can be represented as this part alone corresponding to R0, R1, R2, and this part alone corresponding to R3, R4, right? That will essentially be the same as this. You can test it out. So minus 1, minus 1, minus 1, 0, 1, 0, 0, 0, 1. This corresponds to R0, R1, R2. So we have written that separately. And 0, 0, minus 1, 0, 0, 1. This corresponds to R3 and R4. And so we have written them separately is 0, 0, 0. Now if we transpose this matrix equation, okay, this is an equation consisting of matrices. If we transpose these and pre-multiply both sides by the inverse of the first matrix, you should be a little comfortable with linear algebra to do it. It's a little too difficult to get into those first principles in this lecture. So if you follow it, it's fine. Otherwise, you need to take it on belief and then go and check the matrix algebra and then understand these things better. So if we pre-multiply this matrix with its inverse, this will drop out, and pre-multiply this with the same inverse matrix, this term will remain, and anyway this is 0, so this is going to drop out. Okay. So we have transposed this equation, and therefore all the minus ones have becomes ones, the zeros will remain the same. So this equals this, that's what is our first step, and then we have pre-multiplied it with this matrix, with the matrix that's the inverse of this matrix, which is this. So I inverse is a unity matrix, we are not going to represent that. This matrix times this into R3, R4 will directly give us R0, R1 and R2. Or R3, R4 we are interested in by measuring R0, R1 and R2. No, R3, R4 are the ones that are being measured here, calculated here from the measurements of C and D. With that we can get R0, R1 and R2. So R0, R1 and R2, if you work things out, it will turn out, I mean, this pre-multiplication, this matrix would result in, if you multiply and so on and so forth, R3 plus R4, R3 and R4. Therefore, R0 equals R3 plus R4, R1 equals R3, R2 equals R4. So just by doing this kind of a manipulation, we directly get the intracellular rates based on the extracellular rates that can be measured. That's powerful. Now let us look at nodal rigidity identification. Okay, this is what went into the lysine improvement that we talked about, Valeno and Stephanopoulos' work, early 90s. To understand this, let us take an example. Let us consider the example that popularized the metabolic flux analysis, the lysine production. This is the reference here. It has been also given in your list of references. You can go and take a look at it. Stephanopoulos and Valeno, 1991, Network Rigidity and Metabolic Engineering and Metabolite Overproduction. This was published in Science. And then there was another paper that was published in Biotechnology and Bioengineering in 1993, which gives some more details. As mentioned earlier, L-lysine or minus lysine is produced in annual quantities of hundreds of thousands of tons, because this amino acid is not made by animal cells, but is required for proteins and so on. Therefore, it needs to be provided. That itself causes or gives the reason for the large production of minus lysine. Corynebacterium glutamicum, which is an organism, this produces L-lysine. The yield when this was being produced was only about 15%. And this 15% was increased to more than threefold, to greater than 50%, through metabolic flux analysis. You see the power here? 15% has gone to 50% through metabolic flux analysis and subsequent genetic manipulations. See, metabolic flux analysis gives you directions. Based on that, you need to modify the organism genetically, and then in this case it produced more than threefold yield. So some details. This is the primary pathway that we should be interested in for lysine production. This is a part of glycolysis, glucose going to glucose 6-phosphate to fructose 6-phosphate to phosphoenol pyruvate to pyruvate. And then there is this branch point which is important here, glucose 6-phosphate through ribulose 5-phosphate comes back to fructose 6-phosphate. Phosphoenol pyruvate here goes to oxaloacetic acid and then to lysine. Pyruvate can also go through to lysine, and there is another branch point here from pyruvate to acetyl-CoA. People would recognize that this is the TCA cycle, which is indicated by the dotted circle here, dashed circle. Let us now define a node, which is the metabolite where more than one reaction occurs. So there are three principal nodes or branch points in this network: G6P, where there is branching, more than one reaction occurs, one reaction, second reaction, and then PEP, this goes here into oxaloacetic acid, and pyruvate here, it is going to oxaloacetic acid and L-lysine. Okay. So these are the 3 branch points here. These are the ones that one focuses on to do nodal rigidity analysis. Nodes provide us the possibility of manipulation to preferentially increase the flux through one of the desired branches. Suppose we want the flux to go here to L-lysine, and this is one of the places where a possibility exists to channel the flux more here by some means compared to the flux here. That is what is going to increase L-lysine production, right? So that is why we look at these nodes, the branch points. So in formal terms, if M metabolite undergoes 3 reactions with rates J1, J2 and J3, or fluxes J1, J2 and J3 in this terminology, to give you X, Y and Z, and let us say that Y is the desired product, here J2 needs to be increased selectively over J1 and J3, right? So the split ratio, as it is called, it's the flux through the desired branch divided by the sum of fluxes through all branches. That is a split ratio. It can be written as J2 divided by the sum of all J's, J1, J2, J3 in this case. So J2 by J1 plus J2 plus J3. This is the split ratio. This is fine, but the main reason why this is not so easy is that the flux cannot be changed at will due to the nature of the node. Okay, in all nodes, you just cannot say we'll take more carbon through this branch and stop by probably cutting off the enzyme that is responsible for this branch and so on and so forth. It will not happen. Okay, let me very briefly tell you what it is. There are 2 kinds of nodes that are possible. It could be a rigid node or a flexible node. To do that, let us consider this example. S going to A goes to B and C goes to D and E. These are normalized fluxes. S to A is 100, A to B is 50, and A to C is 50, and C to D is 25, C to E is 25, and some units, millimole per time or whatever, right? So this is the branching when nothing has been done to this particular network. By some genetic means, if we can cut off the enzymes maybe for this branch, then this branch can be cut off, and this branch also is cut off, let us say, then this C to D conversion cannot take place. If it is if the observed fluxes, you cannot really observe it, let us say that the fluxes on doing these 2 cuts turns out to be something like this. Okay. On cutting off these, the 100 becomes 0 here, but it is 100 here. Whereas by cutting off this, both these fluxes have become 0. Okay. Let us say that this has happened. It's not easy to observe this, but let us say this has happened. If this has happened at the node A, it is possible to reroute the entire flux through one branch. Okay, let's say this is the desired branch. By cutting off this branch, we are able to reroute all the flux through this branch, and therefore this is a flexible node. Okay, whereas at node C, because we cut this off, this also got cut off. Okay, we didn't intend to cut this off, but just because we cut off this path, this also got cut off. Such a node C is called a rigid node. We cannot expect to increase the flux preferentially through one of the branch points if the other branch point is cut off. That's a rigid node. So we need to look for flexible nodes to manipulate things, and rigid nodes will not help us. So rigidity could arise due to many reasons. For example, D could be necessary for the branch that is producing E in some way, some metabolic way, some genetic way. It could be responsible, maybe through a feedback loop or something like that. And therefore, if we cut this off, this also gets cut off. So these 3 branch points are shown: G6P, PEP and pyruvate, these 3 nodes. Through experiments and flux analysis, it was found that these 2 nodes, G6P and pyruvate, these are flexible. It is possible to reroute the flux through a desired branch by cutting off the other branch. Whereas PEP is rigid, and rigidity needs to be overcome for better lysine production. You can, in other words, PEP is going to OA, this is the desired branch. This is the PEP carboxylase enzyme that is doing this. PEP carboxylase from the regular organism leads to a rigid node, whereas PEP carboxylase, the enzyme from Pseudomonas, is helpful. Okay, is helpful to break the rigidity. Therefore, the approach was take the Pseudomonas PEP carboxylase, express it in Corynebacterium glutamicum, and that has helped to decrease the rigidity of this node and thereby increase the production of lysine by about threefold. Okay. So, in the last part, we had actually taken the gene from some other organism, expressed it in the host organism. This is the recombinant DNA technology that I was talking about, and this is a way of modifying the cell to produce more. Okay. So, I have shown you an example where the two major aspects, one is looking at the cell in detail and then modifying the cell, have both been shown to improve the yield of a product, in this case L-lysine. And rDNA has been used heavily for insulin production. Insulin is made by using a bacterium. They have taken the insulin gene, or they took the insulin gene and expressed it in bacterium and produced insulin. That was the way by which they could produce large amounts of insulin and bring down the cost and availability and morbidity by significantly. And one more step further, this is changing, putting rDNA, rDNA technology is taking DNA and putting it into another organism. One step further is changing the entire cell. Okay? An example of that is hybridoma. You take a cell that is cancerous, that can grow forever, and you take a cell that can produce a particular type of antibody, put them together, you get a cell that produces only that kind of antibody and can live forever. Okay? That is changing the cell completely. That can also be done to make bioreactors, and if you use them in bioreactors, you can get production of the required product, the monoclonal antibody, from bioreactors. So, in summary, if you look at module 5, we'll summarize the entire course in the next lecture. In module 5, we began by considering the cells themselves, the actual factories that produce the product. We said that we could get some input into what is happening by considering the cell as a black box, and one of the methods of doing that is by stoichiometry, bioreaction stoichiometry, and the degree of reductance concept is an important concept there. Then we said we will open a window and look at it. Some windows are these cell status indicators, the metabolic status indicators, energy status indicators such as redox ratio, the energy ratio, and the intracellular pH. These three are examples, there could be others. And then we said we'll go and we'll go and look inside the cell. That is what we did by metabolic flux analysis through the methods that we have described in detail here, some detail here, not all detail. And then based on the input provided by the metabolic flux analysis, we could do directed change through rDNA technology of the genetic machinery and improve the productivity, for example, L-lysine by more than threefold. And then we said that you could actually change the cell itself by bringing two types of cells together to produce monoclonal antibodies or any other product of interest. Okay, with this we finish the 5 modules. In the next lecture, let me summarize in brief, it will be a brief lecture, what all we did in the course. That will provide you with a nice summary for the course. See you in the next lecture.